Good morning, everybody. Welcome back on this rainy morning. Many of you may know me already, but my name is Ted Rasmussen. I'm a professor in the Department of Pharmaceutical Sciences. And this is the point in this course where we're going to switch gears a bit and shift over essentially to pharmacology, some of the basics of pharmacology and how all of that works. So it's going to be more drug-centered than what you have seen in the past. And this last set of lectures that I will give you are kind of like a self-contained unit, kind of on introductory pharmacology. And we'll use a lot of kind of real-world examples of specific drugs and how they work. So today's topic is really sort of an introduction just to get you all set up. And then and from what you'll see today, you'll notice that when a drug binds to whatever its target is, might be a receptor, might be something else, how tightly it binds is very important. So on Wednesday, we'll get into a little bit of the biochemistry and quantitative ways that we can look at how tightly the affinity of a drug is for its target. And then on Friday, we're going to talk a little bit about selectivity, which is how a drug might bind a number of different targets with different strengths, basically. But anyway, we'll get going. And you can find my email on the syllabus. And if you want some additional help, feel free to shoot me an email and we'll find some time to meet. Another thing I will try to do is be available to stick around for a little while after each lecture. So I welcome you to come up and discuss further whatever you want after we're finished. And I also welcome questions. I mean, if there's something you really want to address right in class, I would really encourage that. Just raise your hand and we'll take questions on the fly. Okay? Is my volume okay in this room with this microphone? Not too loud, faint? Okay, good. So we'll go ahead and get into this. I'm going to start off very basic. What is a drug? And if you think about it, a drug is any exogenous substance that you would put into your body. And of course, it would exert some sort of a physiological effect when introduced into your body. Now, the drug could be of natural origin. It could be extracted from plants or animals, for instance, or it could be synthetic. But the idea is it is what we would call a xenobiotic, meaning something that you ordinarily would not encounter, but it's intentionally introduced into your system. And the idea is it's going to exert some sort of a beneficial or a therapeutic effect. If it were something that exerted a negative effect, we would call it a toxin. And actually, you'll get a sense for this as we go along, that we want the benefits to outweigh the risks of a drug. And all drugs have certain side effects, but they vary greatly in the severity. And we want the effectiveness of the drug to be much greater than the potential toxicity. And we also, if you think about like the FDA, they're also interested in the severity of the disease that they're treating, right? So you might tolerate some negative side effects for a cancer therapy, but you wouldn't want many negative side effects for like a topical ointment that reduces itching, for instance. So kind of common sense. And what is pharmacology? Well, it's simply the branch of medicine and of science that is concerned with the effects and the modes of action, MOAs, of drugs and the safety of drugs. So you'll see this word, or this three-letter acronym, MOA, all the time. That's the mode of action. Sometimes it refers to the mechanism of action, but that is how the drug actually works at a biochemical level. Okay, so let's get into this a bit more. And I want to introduce right now two of the major subdivisions in pharmacology, which are pharmacokinetics and pharmacodynamics, which we call PK and PD. They're really different approaches, and to fully understand how a drug works, you need to understand that drug's pharmacokinetics and also its pharmacodynamics. So you can kind of figure out a little bit from just taking apart these words. Pharmaco means drug, right? Kinetics means in real time how the drug accumulates, for instance, in the bloodstream or at its target site, what affects the amount of drug that's delivered, and what happens to it after it's delivered, where it goes, these types of things. Whereas the word dynamics and pharmacodynamics really more addresses how it works. So the MOA, for instance, is a pharmacodynamics consideration. How fast a drug is absorbed after you take a pill is a pharmacokinetics consideration. So a little bit more about this. So broadly speaking, we can, and by the way, I am recording all my lectures and they'll be posted on Husky CT typically the same day. So that hopefully makes it a little easier for you to just listen to what I'm saying, and you can go and review all of this after the fact. Okay, so pharmacokinetics. Basically, what is the concentration of the drug after it's administered, and also how that varies over time. You know, how quickly does it get into your system? What levels does it build up to? And how long does it persist? And so that's all a pharmacokinetics concern. Basically, what is the concentration over time after given a single dose? What's the disposition of the drug after administration? And by disposition, I mean, where does it go? Another important thing is what's the half-life of the drug in your system? And typically these things kind of exponentially decay. It will build up to a concentration, and then after a certain period of time, you'll have half as much. And after another period of time, you might have half as much as that. So now you've got a quarter of it, and then half as much after the next unit of time. Now you're at an eighth. And so these can be, pharmacokinetics considerations can be described very mathematically, and they can also be determined in a laboratory setting or in a clinical setting very easily by withdrawing blood samples at discrete times and just simply taking that blood sample and measuring how much of the drug remains. So pharmacokinetics is all about the amount of drug, how long it persists, where it goes. And if you see this, basically what happens, the drug is administered, and of course drugs can be administered in different ways. It could be orally available, it could be in a pill or a tablet, which is swallowed, and then it would have to be absorbed, typically through the lining of the small intestine, and then it enters the bloodstream, right? And then it can be distributed throughout the body. So how quickly it's absorbed, how quickly it gets into the bloodstream, how long it persists in the bloodstream, that's important. But equally as important as how it builds up in the blood is how it's removed. And so the drug can be metabolized, its chemical structure can be changed by enzymes in the body, and most of the important enzymes are divided into, incidentally, enzymes called phase 1 and phase 2 enzymes. And phase 1 enzymes are cytochrome P450s, CYP enzymes. Humans have about 70 or 80 of them, and they make small molecule changes to the drugs. And so the drug might have to be metabolized in order to be excreted, or maybe the parent drug is the active form, and it gets excreted without much metabolism. It depends on the drug. It's also important how it gets to different tissues, how the drug is distributed after you take the drug. So all of these considerations are embedded in this term ADME, and we'll see that in more detail on the next slide. But ADME is very important, and the A stands for absorption, the D stands for distribution, the M stands for metabolism, and the E is for excretion. So think about it. I want you to definitely be thinking that it's important how the drug gets absorbed, how it's distributed throughout the body, how it reaches the bloodstream, what levels it builds up to. But equally important is how quickly is that drug metabolized into something else, and how is it excreted. There is a bit of a an important consideration here too, the metabolism part, and that's whether the drug needs to be bioactivated. So some drugs that we take are in the form of parent drugs, and the parent drug is whatever is in the formulation, but that particular drug might not be biologically active. What has to happen is it has to be activated by metabolism in your body, and then the metabolites are actually the active drug. So bioactivation kind of comes into this too. Okay. And there are other drugs where the parent drug is the active drug, and then metabolism is involved in changing it to a different structure that is then subject to, that can be excreted. So it depends on the particulars of an individual drug. But I want you to kind of burn the concept ADME into your minds, dream about it, do all that stuff. It's a really important term. And all of that is in the realm of pharmacokinetics, which is kind of the upper part of this diagram. Pharmacodynamics, in contrast, really has much more to do with how the drug actually works at a molecular level, or at a cellular level, or a physiological level. So for instance, you might take we would be interested in what is the drug concentration at the site of action. So many drugs are just systemic, right? If you take an aspirin, the aspirin drug goes to all parts of your body. But you might have arthritis or something like that, or you might, whatever you want to use that drug for. The same is true of maybe Tylenol or whatever. Tylenol kind of tends to work somewhat in the brain, but anti-inflammatories, NSAIDs like Motrin, Advil. We're interested in how much of the drug gets to the place where the pain is, or the neural circuits that regulate pain. But it's going to be present in all your tissues as well, not necessarily doing things there. So pharmacodynamics, obviously, we're interested in where the drug is, which kind of is an outgrowth of the ADME concern. But then most importantly, we want to know the pharmacological effect. What does it do? What are the targets of that drug? Typically, they're discrete entities in our body that the drug binds to, like a receptor or an enzyme or a structural protein, that type of thing. And so that's the target of the drug. And so how that drug molecule interacts with its intended target molecule is very important and in the realm of pharmacodynamics. Then, of course, we want to know what is the clinical response. Some drug maybe binds to a particular receptor, and of course, receptors allow cells to respond to their environment, essentially. And so when something binds to a receptor, perhaps for a drug, maybe that cell changes what that cell is doing. Maybe it causes it to open and close channels, and ions go in and out of the cell, for instance. Or maybe it causes the cell to change shape. Or if it's a muscle cell, maybe it causes it to contract or relax. So what is cellular physiology that's elicited by that receptor? And the drug activates, or perhaps in some cases inhibits that receptor. So this is really kind of the clinical response, the medical response. And of course, we're interested in these two terms, efficacy and toxicity. So efficacy means how well does the drug work to do its intended job. And we'll get into we'll spend another lecture entirely on that topic really later on in a couple of weeks. But how well does the drug work? How does it work and how well does the drug work are both in the realm of pharmacodynamics. But we also want to understand the toxicity of that drug. And most drugs have beneficial, efficacious aspects to their action, but they also have unwanted side effects, adverse events that can possibly happen. And so we want to understand that as well. So the two broad fields of pharmacology, pharmacodynamics or pharmacokinetics and pharmacodynamics, PK and PD and ADME. So you're already getting all of these acronyms. Okay, questions so far? Okay. Now we're going to focus in on each of them in a little bit more detail. So pharmacokinetics. It's basically, one aspect of it is the study of how drug concentrations change with time after you administer the drug, how long and where it persists in the body over time. And as I mentioned, these parameters are all affected by ADME, which is absorption, distribution, metabolism, and excretion. So you'll see a lot of kind of curves like this that are derived from real world experimentation, oftentimes in animals first and then in humans during clinical trials. And typically, what we want to know, just at first estimate, is if you just give one dose of the drug, what's going to happen over time. So on the x-axis of this is time, and on the y-axis is the amount of the drug. And oftentimes this is measured in blood because blood is very easy to get, right? And so what would happen is, if it's a lab animal or a human, you administer the drug, and then at discrete time intervals, you withdraw blood samples and you measure how much drug is in the bloodstream. And typically, you can see that the drug, of course, it's not present before it's administered, but as soon as it's administered or soon thereafter, it will start to accumulate in the blood and build up, and then it will decline. And basically, the first part of this is mostly driven by absorption, and then the last half of this curve is due to elimination. And so here we're following the drug building up. Maybe this is like after an hour, it reaches a maximum value. We would call that the Cmax value, the maximum concentration that given amount of drug reaches in the bloodstream. And then it starts to decline. And that's because typically it's being metabolized and excreted. And one very important measure of this is the half-life, the T1/2. This is the amount of time it takes for the Cmax, the maximal concentration, to be reduced by a factor of 2. There's half as much after one half-life. And then there's a fourth after 2 half-lives, et cetera, in a perfect world. And then you could even define Cmin, the lowest concentration of the drug in the blood. Oftentimes that's 0. So one thing you'll notice is that this is not really a perfect curve. In this case, it builds up rather rapidly, and that takes a long time to decay. And that's because the buildup is driven by absorption and distribution to some degree. And the elimination is driven by how fast it's metabolized and also how fast it's excreted. So many drugs are excreted through the kidney, for instance, okay, and eliminated into the urine. But other drugs, for instance, might be metabolized in the liver and put into the bile and then into the digestive tract, and elimination might be through feces. And so those have different kinetic considerations. So the mechanism whereby it's excreted is also affecting all of this, okay? So another thing I want you to think about is, if you want to look at the total amount of drug that's delivered over time, this can be described by area under the curve, AUC. So if you look at this, if you use some math, which typically involves some calculus actually, you can determine the absolute amount of drug that the body is ever exposed to by looking at the area under this curve, right? And that gives you an idea of how much of that drug is actually delivered into the bloodstream. If you think about how drugs are prescribed, they might say, take 2 pills per day, right? Well, most likely what they're doing, the first pill gives you a dosage like this, and maybe once you're down in here, you take a second one. But if you get the dosages close together, you have 2 areas under the curve, one for the first dose, one for the second dose, and the amount that's present is going to be additive, right? So you'll have to add areas of curves together. And it's worth thinking about that when you have repeated dosing, you have kind of like a spike wave. It builds up, starts to decline, and you take another dose, it builds up, starts to decline, etc. And so it's very difficult to dose people with a uniform level of drug over time. You always have this kind of spike wave sort of response with repeated dosing. And if you dose too close together, since it's additive, it will start to build up, perhaps to too high of a level. So that's also a very important pharmacokinetic consideration, is multiple dosing. Okay, pharmacodynamics, how the drug works, the MOA, what's the mechanism at the molecular, cellular, and physiological levels. And it's really kind of the study of the relationship between drug concentration and the response that you're going to have to that drug. So pharmacodynamics is much more biological. So for instance, you can look at the ligand-target interaction. So the target is the molecule that the drug binds to, the intended target, and the ligand is anything that binds to that target. Oftentimes it would be a drug. Sometimes it's an endogenous ligand, a ligand that's built, that your body produces. So you can have oftentimes competitions between a drug and an endogenous ligand, maybe some kind of a hormone, for instance. And there can be a competition for binding between the drug and the endogenous ligand. But at any rate, how that drug actually binds physically and molecularly to its target is very important. So this is kind of a space-filling model of a protein with some key atoms shown, and it's a protein, so you have amino acids in here that are numbered based on where they appear in the protein. And then here is a drug in the middle. And there are interactions, molecular interactions between the drug and its target. Usually these are non-covalent interactions. There's no physical binding, but they're controlled by things like hydrogen bonding, van der Waals forces, things like this. But the drug oftentimes fits into a little pocket because it's designed to do so. And so using crystal structures, which is basically determining exactly how that protein is laid out and how the drug interacts with that, is in the realm of pharmacodynamics, and we would call that, a structural chemistry sort of view of a ligand-target interaction. Of course, the structure of the chemical structure of that drug is very important because it has to interact somehow in some sort of an exquisite kind of lock and key mechanism with the target. But once that drug binds that target, well, that's great, but what does that cause? Well, it may cause a cellular change. And so here we have to consider what are all of the cellular responses that the cell experiences after the drug binds. Typically, what you're doing is tricking a receptor into doing something that it ordinarily would do in a cell by binding a drug to it. Or the cell is doing something we don't want it to do, so we block a receptor with a drug so that it doesn't signal. And those are the two general themes. And broadly, that's agonists and antagonists. So to fully understand a drug action, we really have to consider many levels. And it has to do with essentially biology. We have to consider all of the levels that go on in the drug-organism interaction. What is the molecular structure of the drug or compound? How does it interact with its target? And then how does it affect organelles within the cell itself? Does it affect tissue organization or tissue function? And ultimately, of course, that's going to affect the organ itself. And then we have organ system and organism. So we really want to understand the biology all the way from the molecular level all the way through the physiological level, and then how it affects the whole patient. Okay. All right. So target engagement is important. And so if you think about kind of what happens, it's true that most drugs act at a molecular level, and that's going to influence the cell in some way and cause some sort of a beneficial effect, hopefully. And hopefully it's going to cause a beneficial effect that's relevant for treating a disease or a disorder. So in this course, you're just going to see over and over in what I teach how the drug affects the target itself, the ligand-target interaction, and most typically, an example is the ligand can be a drug, and the target might be a receptor, or it might be something else. So that's the first consideration. Then what's the molecular response to that drug-target interaction, the ligand-target interaction? How does that molecular response feed into and affect cellular responses. And I should also say probably physiological responses. And then how is that change going to affect the course of a disease? You know, what's the medical effect of it? Okay, so I want to just change gears a bit and talk about receptors, because receptors are the most common drug targets. They're not the only drug target, but they're very commonly the drug target. And so let's talk a little bit about receptors. You've already heard a lot about receptors in this course so far and elsewhere in your education, but just to review, most receptors are embedded in the cell membrane, and they're there to do something, right? They're there to signal the cell to do something when something binds to that receptor. We didn't evolve receptors so that we could develop pharmacology, right? They're all there naturally occurring, and receptors bind something endogenous in your body normally. And so what we're really doing with pharmacology is we're tricking that receptor with an exogenous compound, a drug. So most of them are in the cell membrane, but there are some receptors that are actually intracellular or even nuclear. The receptor binds to a ligand, which could be an endogenous normal compound like a hormone or some such, or neurotransmitter is another great example of an endogenous ligand in terms of neurobiology. But the drug can also bind the receptor, and that initiates some sort of cellular response. And again, that's going to affect the physiology, the health of the individual. So looking at receptors in a little more detail, there are 4 major classes of receptors. Some of them are actually kind of receptors and channels. And this is an example, a ligand-gated ion channel. So this is a channel that's in the cell membrane, and ions can go in and out, in this case sodium, okay? And that could regulate muscle contractility, neurotransmission, things like this. But some of these channels also have receptor-like features to them. For instance, a drug might also bind nearby and regulate, or an endogenous ligand, and regulate the opening and closing of that. So it's kind of a weird hybrid between a receptor and a channel. Another major class of receptors are GPCRs, G-protein coupled receptors. And we're going to have actually 2 full lectures on these because they are the most common drug target in general. And these are interesting receptors. They're embedded in the plasma membrane, not shown here, but they also have these internal components called G proteins. So you have a receptor that cooperates with something called G proteins. And when an agonist binds these receptors, they usually have 7 transmembrane domains, it will activate a G protein, and the G protein goes along and activates second messenger systems, like it might make cyclic AMP, for instance. And then these go on and through the process of signal transduction, activate the cell to do something. And so GPCRs are a very important type of receptor, and they're somewhere near, oh gosh, probably upwards of 1,000 of these in the human genome. So they're very common receptors. You also have tyrosine kinase receptors. These tend to be dimers. Growth hormone receptors are a great example of this, for instance. And instead of having 7 transmembrane domains, they have only 1. And oftentimes when an agonist binds one of these, they cause it to dimerize. And so when it's dimerized, then it kicks off kinase activity. So it will phosphorylate tyrosine, for instance. And there's sometimes autophosphorylation that goes on. And that then goes on and tells the cell to do something. And then the last type of receptors are called nuclear receptors, and they're very strange receptors because they aren't present in the plasma membrane. Rather, they can be present in the cytoplasm. And in this case, the drug has to or is capable of diffusing right through the plasma membrane, and it encounters a receptor in the cytoplasm. And then typically, when that happens, the drug receptor complex translocates into the nucleus. And the weird thing about nuclear receptors is they're also transcription factors. So it will then, what's in the nucleus? That's where the DNA is, right? So that nuclear receptor can then act as a transcription factor and start transcribing target genes, and so you get a genetic response, okay? There are some nuclear receptors that are obligately in the nucleus, and you have ligands that can go all the way through the cell membrane, through the nuclear membrane, and then it encounters the receptor in the nucleus itself. But these are hybrids between receptors and transcription factors, and many of them can move from the cytoplasm into the nucleus. So those are kind of 4 major types of receptors. The first one's weird because it's kind of a channel/receptor. But these are often drug targets. And you'll see here it also says agonists. So agonists are drugs that actually activate the receptor. You could also have drugs called antagonists that inhibit the receptor. And whether you want an agonist or an antagonist depends on the biology. Whether it's beneficial to activate that pathway or whether it's beneficial to inhibit that pathway. So there are, I talked about receptors just now, but there are also a lot of non-receptor drug targets. Nucleic acids, some drugs target DNA and RNA directly. Some drugs can target structural proteins, things like tubulin, actin, that type of thing. Some drugs can target enzymes, the proteins that actually do chemical reactions, right? Some drugs target ion channels, and others are regulating cytokines, which are endogenous ligands that do something in the body. So I would say most drugs target receptors, but many do not. And so these are some examples of these. So receptors, ion channels, enzymes, and I'll introduce transporters as well. So, an agonist is something that activates a receptor, and it binds to a receptor, and then it does something. Things like it opens and closes ion channels. That receptor might cause an enzyme to become active. It might modulate an ion channel. It could cause DNA transcription, like if that's a nuclear receptor, for instance. An antagonist would block that receptor, and you would have no effect. So all those pathways would be prevented from happening. And sometimes that's very beneficial to prevent the body from doing things. Ion channels. Some drugs actually just block ion channels. They plug them up by binding to the place in the channel that ordinarily conducts ions. And that would block ion transport. Other drugs can bind elsewhere on the ion channel and modulate it. It means it changes its propensity, the number of ions that will flow through per minute, for instance. Enzymes. You can have enzyme inhibitors where the drug actually prevents that enzyme from working. Some drugs are actually false substrates. So substrate is whatever that enzyme works on. Remember from like biochemistry, a substrate gets turned into a product. So the substrate is pre-metabolism, the product is post-metabolism, and the enzyme is doing the metabolism. So if you have a drug that looks like the normal substrate of an enzyme, then there's going to be a competition between the false substrate, which is a drug, and the normal substrate. And if you have a competition going on, the rate at which that enzyme makes product is going to be reduced. And then, of course, I want to mention that some enzymes, especially CYP enzymes, are actually involved in drug metabolism. So they can convert a prodrug into an active drug. In other words, metabolism is needed to activate that drug. Cells also contain transporters. And these are somewhat similar to ion channels because they move things across the membrane. But a lot of drugs get into the body via transporters or into the cell. And then there are a lot of transporters that pump drugs back out of the cell. And so drugs can affect ion channels, or excuse me, transporters in kind of similar ways. Here's a normal transport, a drug, or an entity, for instance, some kind of a ligand binds to that transporter, and then it gets moved across the membrane via that transport. Well, if you have an inhibitor, it will block that transporter, and this purple ball here can't get in, right? You can even have false substrates where you get an unusual compound that gets accumulated. But anyway, you get the idea that there are a lot of things besides receptors that can be drug targets. Okay. And lastly, there are a few things that have non-molecular mechanisms. So for instance, alkali or acid burns, that's not really a particular target, it's just a feature of an acid or a base. You can have drugs that act by chemical antagonism. For instance, antacids just alter the pH in your stomach. Doesn't really have a target, it's just going to directly change the pH because it's a buffer, for instance. Chelators are drugs that bind other things and just take them out of action. They sequester them. They bind it and take it out of action. Some drugs, osmotic diuretics, for instance, change the water balance across membranes, and they can cause elimination of excess water. Some drugs just disrupt membranes in general. So these are kind of non-molecular mechanisms, but these are kind of rare as drugs. Okay, and of course, since there are so many drugs, there are a lot of classification schemes for drugs. Drugs oftentimes come in families. Receptors do too, by the way. And we tend to categorize and classify drugs based on their structural characteristics or the signaling pathway they affect, or sometimes by the level of organization, whether it's the molecule, the cell, the tissue, et cetera. So drug classification schemes are kind of all over the place, but they're useful because oftentimes, there are whole families of drugs that do similar things. Okay, so the last thing I want to do in the remaining 14 minutes here or so is zero in on one part of this, basically pharmacodynamics, and that is the relationship between the chemical structure of the drug and its activity. So obviously drugs have very discrete chemical structures, and that's very important. So the study of this area is usually an attempt to determine something called an SAR, a structure-activity relationship. And if you think about that word, the structure is what is the chemical structure of the drug, and given that chemical structure, how does that structure affect the activity of the drug? Okay, how well it works, basically. So an SAR, structure-activity relationship, is simply the relationship between the chemical structure of a molecule, a drug, or even an endogenous ligand, and its biological activity. So what we're really interested in is, so we have to consider the chemical structure of the drug, but we also have to consider the chemical structure of the target to some degree, because they interact, right? Okay, so if you think about the chemical structure of the drug, it's just a chemical structure. What happens, or how important are particular bonds, covalent bonds in that drug, the composition of the drug, how that molecule is organized. And typically, most drugs interact with their targets by non-covalent interactions. So for instance, there's oftentimes, usually there is not a chemical reaction between the drug and its target. If there were, that would mean the drug would physically form a classic covalent chemical bond with its target. These are usually non-covalent. And so they're driven by weaker forces of interaction, things like hydrogen bonds, hydrophobic interactions, charge interactions, things like this. Local charges within the across bonds within the drug, dipole moments. So these are the types of kind of weaker interactions that we're going to be thinking about. And probably some of the most important ones are charge interactions, hydrogen bonding, that sort of thing. But if you put all that together, we can kind of think, what is the molecular interaction potential of a drug, given that we know what its target might be? Okay. So the structure of the drug can affect all sorts of stuff. So, here's kind of a cartoon of drugs. A drug-like scaffold is a chemical structure that could be modified and played around with by a pharmaceutical company, for instance, to try to change its activity. What would changing the structure of the drug subtly do? Well, it might affect the affinity for a target interaction. It could affect the receptor-drug interaction, obviously, which is kind of similar. It could alter the selectivity of the drug. So, for instance, there are a whole bunch of estrogen receptors. We're going to talk about this some on Friday. There are a whole bunch of estrogen receptors, and there are a whole bunch of estrogen-like drugs. Some drugs will bind all the estrogen receptors, all 8 or 10 or 12 of them. Some of them will bind only one. And so that's a selectivity consideration. And oftentimes we want selectivity because you'll have less side effects if you have a very targeted interaction with one receptor. The structure of the drug can affect the toxicity. So maybe you can tweak the structure of the drug and reduce toxicity. Maybe the structure of the drug can affect pharmacokinetics or formulation considerations. Maybe you can make it water soluble instead of hydrophobic, and you can turn it into a pill form more easily. The modifications can modulate metabolism, maybe affect bioavailability, how well it's absorbed. And so these structure features of the drug are affecting all aspects of ADME and pharmacokinetics and pharmacodynamics. And then some pharmaceutical companies do something called scaffold hopping. Somebody makes a great drug and they tweak one methyl group, has no effect on the drug except it makes it into a new chemical entity and it can be patented. So that's kind of a little bit weird, but happens all the time. So here's an example. I want to show you one example, and it's an example of corticosteroids. Okay, and we're going to look at the structure of this and see how tweaking different components of the chemical structure affect the activity and behavior of this drug. So the base of corticosteroids is a 4-membered sterile ring structure, which you can see in the center here. And you can see that the rings are called A, B, C, and D. It's 6, 6, 6, and 5-member rings, and that's the structure of all steroids. And you can see here that by changing different chemical features, it can affect corticosteroids. So there are two major types of corticosteroids, mineral corticoids and glucocorticoids. The mineralocorticoids regulate salt and water balance. Glucocorticoids regulate blood sugars, stress, and inflammation. So they do slightly different things. But look at what happens when you do this. A hydroxyl group placed at position 11, that's an absolute requirement for glucocorticoid activity. So if it's a glucocorticoid, you have to have a hydroxyl group here. If it's not present, it's no longer a glucocorticoid. You can just kind of randomly go around here. Increasing a double or adding a double bond right here in ring A increases glucocorticoid activity. But it also slows metabolism of the drug, okay? In the case of like prednisone. And so if you alter the metabolism of a drug, that's going to affect pharmacokinetics, right? If you slow the metabolism of the drug, that drug will persist longer. So by tweaking that double bond right there, you can make that persist in the body much longer. Here's some things that regulate corticosteroid activity. You can see all sorts of things in here. This is an interesting one. If this is esterified right here, makes a lipophilic molecule that's good for topical corticoids. Out here, hydroxyl group right here is an absolute requirement for mineralocorticoid activity. And so these are subtle changes in the structure, but they have effects on how well the drug works, what kind of drug it is, how long it persists in the body, and all sorts of these things are done. All right, so, and remember that, we're talking structure-activity relationships. This slide here is just focusing on the chemical structure of the drug itself, right? Remember, though, that this is probably binding to some receptor, and so those chemical changes in the drug are going to influence how that drug interacts with its target, a receptor, for instance. And so if you want to know why adding a hydroxyl group here is important, it's probably because it's changing the way that drug chemically or interacts with its target. Might affect a hydrogen bond or something like that, okay? But these are affecting the efficacy of the drug, kind of the selectivity of the drug. In this case, the selectivity is it a mineralocorticoid or a glucocorticoid? Maybe some of them are sloppy and they're kind of both, So you could, they're also affecting things that affect ADME, and they're also affecting certain things that affect the formulation of the drug, whether it's hydrophobic, hydrophilic, that sort of a thing. And so to get a full picture, we'd also have to have a look at the structure of the target as it interacts with each of the structural components. One thing I would like to say really quickly right now has to do with artificial intelligence. So pharmaceutical companies are really interested in AI right now, okay? And that's because there are libraries of potential drug compounds that number, where the components of each library numbers in the millions. So, in our hands we have millions of compounds whose structures are known that could potentially be new drugs, okay? But, it's very difficult in the lab. Back in the old days, you would just start plugging away and start testing this drug number 1, drug number 2, drug number 3, what does it do, what does it do, what does it do? But with artificial intelligence now, AI platforms can actually predict favorable structures, or which types of chemical structures cause predicted better interactions with targets. And so it's not the total answer to everything, but artificial intelligence can rapidly screen through, once it's properly trained, and screen through millions of potential chemical structures and find the 4 or 5 or 6 compounds that are potentially an excellent fit to a particular target. And then you can go test it out in real life, but that's a very powerful thing, isn't it? AI models can also be loaded up with all of the different targets and information on what those targets do, what the receptors do. AI models can also look at diseases and understand what are the most important pathways in that disease, and what are the what are the receptors involved in that disease, and what compounds might interact with those receptors. And so they can really crunch through all of this complexity fairly quickly. And so I think there's kind of going to be a new wave of AI-generated drug candidates. And most of the big pharmaceutical companies are doing this now. In fact, there are a couple of companies out there that are exclusively doing this as a service for the larger pharmaceutical companies. So you'll be seeing more of this. All right. So in summary, you know that drugs act through macromolecular targets, interactions, and they have to influence some kind of a cellular response, and the structure of the drug is very important. So what we'll get into on Wednesday is we're going to start talking about the affinities of drugs for their targets, which will get a little bit more mathematical and biochemical. But anyway, that's a pretty good introduction, a little bit of a changing gears, and I'll stick around for a few moments, and if you have questions, feel free to come up and ask them. All right, everybody stay dry out there.